All right, welcome back to biology. We've been talking about the cell cycle and reproduction, Unit 7. Remember, I told you that the last set of notes was broken up into two sections. This is Section 2. Hopefully, we can get everything in our allotted time, but we're going to jump right into it. So, we finished up talking about asexual reproduction and your cells, how you grow. We learned that your cells reproduce by something called asexual reproduction. They go through the cell cycle. The cell cycle is just the time in between each cell division, the time it takes from one cell to go from dividing to the next time it divides. Okay, this is of the cell cycle that you need to know. The first one's called interphase. It has three parts: G1, S, and G2. We'll talk about what those are here in a little bit. There's a phase called mitosis, or the M phase, when the nucleus divides. Okay, it's important to know that mitosis does not mean cell division. Mitosis is when the nucleus from one nucleus to two. And then the last phase of the cell cycle before we start interphase over again is known as cytokinesis, and that's when the cytoplasm actually divides and we get two new daughter cells on either side. Okay, if I was going to draw a picture of the cell cycle, it would look something like this. You can see interphase is a long period of time. The cell really doesn't do dividing all the time. It spends most of its time just kind of hanging out and going through these other phases. In fact, there's even gaps and pauses in between each one of these phases so that the cell doesn't keep going through this cycle constantly. If it would do that, that would be known as cancerous, and we'll talk more about that here in a second. But you can see the interphase is the longest. You can see all the hours that we spend in each of the G1, S, and G2. We'll talk about those here again, like I said, in a second. And then there's the M phase that's also known as PMAT, and we'll talk about what PMAT means. But maybe you can tell me what's wrong with this picture. Okay, well, what's wrong with this picture is that, remember, mitosis, we said, doesn't mean that the cell actually divides. That doesn't occur until after telophase and before G1 during a process called cytokinesis. So this little extra picture in the bottom is incorrect. That would actually only occur in this last phase. Okay. So interphase is what it looks like. It just looks like normal cells. Nothing's really happening. We're not dividing, but we are preparing to divide. We're always doing that. Okay. We said it's made up of three phases. G1 is known as the growth phase, or the first growth. Right after you're born and you make that daughter cell, it has to get bigger. Remember, it's half of an original cell, so we're going to have to grow to a normal cell size. So that's what the G1 is for. The S synthesis, that means DNA replication is taking place. Now, why would we want to replicate our DNA? Think about what the advantage of that would be. Well, that means we're about to make a new cell. We're getting ready for that cell division. So that's what the S phase is for. And then G2, this is when all of our organelles are going to duplicate. We're going to grow a little bit in size to get ready for our division to be split in half. So growth stands as what the G stands for. Synthesis is what the S stands for. So how do cells divide? They go through a process beginning with mitosis. And I remember mitosis as PMAT. Everybody uses their PMAT. I know that other people learn that I pray mostly at the church and stuff like that in order to remember interphase, prophase, metaphase, anaphase, telophase, and cytokinesis. Whatever you want to remember it as, that's fine. But I just remember I use my PMAT. Prophase, metaphase, anaphase, telophase. Okay, let's talk about what those mean. Now, you can see you're going to have to be able to identify some pictures of each one of the phases. And they each have their own unique markers or identifiers that help you find each one of the phases. And we'll talk about each one of those as we go through the notes here. So prophase, the first stage of mitosis. What's going to happen is you can see our nucleuses on either side, or nuclei, they're starting to disappear. We remember them as being a big, fat, dark circle. Well, now those dark circles really aren't there. And in fact, you can even see some gaps in the edges of those circles. That's because the nuclear envelope, the nuclear membrane, is starting to break down. It's not there anymore. And then the chromatin, which made up the dark red area, which is, remember, the DNA and protein combined, they're now actually bundling into the chromosomes. So you can start to see the chromosomes actually appearing here. What's harder to see is the centrioles that are moving to opposite ends of the cell, and then the spindle fibers that are attaching to the centromeres. And if you remember from our notes the other day, centromeres, that's the center of that duplicated chromosome. Okay, that X, it's the middle of that X. That's the centromere. So spindle fibers are attaching to those. Metaphase is the easiest one to identify because metaphase means middle. The chromosomes, the sister chromatids, are lining up, the duplicated chromosomes are lining up in the middle, and they're preparing to be pulled apart in opposite directions. Okay, that middle area, we know that as the metaphase plate. It's the middle of the cell. It's where we're preparing for metaphase. And then the spindle fibers are usually present. You can kind of see them here in the blue. You can kind of see some lines attaching to the duplicated chromosomes. But here they're really easy to see. You can see those spindle fibers. The center, that little dark spot in the center, that's the centriole. And the way I remember it, or the analogy I like to give is the centriole is like a fisherman, and the spindle fiber is his fishing line. And he's trying to catch him a chromosome. And what's he going to do with that chromosome when he catches it? Same thing a fisherman would do. He's going to reel it in. And that's what anaphase is. The spindle fibers are shortened just like you would on a fishing reel. You're going to shorten them, and then it's going to pull those sister chromatids apart. It's going to pull them to opposite sides of the cell, and that's what anaphase looks like. So metaphase, they line up in the middle. Anaphase, they're pulling apart. Telophase, then, now they're on opposite sides. Now they're going to unbundle. They're going to uncoil themselves back, and they're going to start to look like a nucleus again. The chromosomes are going to disappear, but the chromatin is still there, and that's what the darker circles are. Okay, the spindles are no longer needed, so they kind of dissolve, and then the cytokinesis can actually start to happen. You can see cytokinesis beginning in each of those. And now you say, what is cytokinesis? Well, cytokinesis is the division of the cytoplasm, hence the name cytokinesis. Okay, there are two different types of cytokinesis, whether you're a plant or an animal. Remember, animal cells are like more squishier, they're more flexible, so they're real easy. Like if this is my original animal cell, I can just kind of squeeze in the middle and pinch it apart, and now I got two new cells. Plant cells can't do that. Okay, and we'll talk about what they do here in a second. So animal cells make what's called a cleavage furrow. Okay, they cleave, meaning that they split in the middle. Okay, so that space where my fingers meet, that would be the cleavage furrow, and then they split at that cleavage furrow. Plant cells, since they have a cell wall, they can't bend. There's no bending to them. Remember, we said that makes them inflexible. That gives them their rigidity that holds them up. So what they have to do is right in between the two, they're going to form what's known as a cell plate, which is going to become the new cell wall in between the two new cells. Okay, this is a reason that plant cells don't need the centrioles the way that the animal cells do. Centrioles not only pull those spindle fibers across, but they also pull the cell apart. Now, you don't have to pull a cell apart in the plant cell, so they don't need those centrioles. They don't need the fisherman. They just need the fishing line. Okay, take a second, pause this, see if you can figure out which one of these 5 goes into which phase, and then I'll come back and I'll show you which one it is. Ready, pause, and go. Okay, so interphase, what am I saying? Interphase is an easy one to identify because there's a regular nucleus. Prophase, remember we said the chromosomes are starting to form, so you can see that in this phase. Metaphase, we said, is the easiest to identify because they line up in the middle, and then they pull apart during anaphase, and then we get 2 new cells during a process called telophase. 2 new nuclei, excuse me. Okay, so what's the result? The result is I get 2 new daughter cells with a complete set of DNA on either side. Okay. All right, how do cells control that growth? What do they do to control how often they're replicating? Okay, well, they generally, in a normal cell, should take about 20 hours to do one cell division to the next. So you spend most of that time, again, in interphase. Your brain and heart cells hardly ever divide. In fact, most brain cells, when they're gone, they're gone. Skin and digestive tract cells, they reproduce all the time. You are constantly making new skin and hair, and you're making a new layer of mucus and cells in your stomach to help protect them. So if they need to be replaced often, they're going to be replaced often. Your body is smart enough to do those types of things. So some cells will reproduce faster than others. Okay, embryonic cells, when your first embryo, remember an embryo is the first when the sperm meets egg and you got that fertilized egg, when it starts to make that first clump of cells, that's known as an embryo. Embryos form into blastocysts, which is like the little ball of cells, and then we have another inner set of cells on the inside that's going to become an adult or a fetus at some point. And those blastocysts and embryo, remember, those are stem cells, so they can become any kind of cell. So since they're stem cells, they're going to need to reproduce quickly and often, so we can start to make that baby, make that fetus. So it takes them about 30 minutes to go through one cell division to the next. Okay, cyclin and regulatory protein. Cyclin is a protein that controls the cell cycle. It tells the cell when to pause, when to stop, and when to go. Okay, and regulatory proteins, like we talked about with the cut earlier, like external factors might be something like if I'm not touching another cell, then I'm going to keep growing until I do. That might be an external regulatory factor that controls cell division and growth. Now, if there's something wrong with that process, if for some reason the cyclin or the regulatory proteins and factors aren't affecting the cell correctly, that's known as cancer. And cancer is caused by something called a tumor. Now, a tumor is a group of cells that are going through uncontrolled cell division. You go from 1 to thousands without stopping that process. Remember, we said there's supposed to be pauses in the cell cycle. There's no pauses in cancer cells. Okay. There's 2 different types of tumors. There's benign tumors, meaning that they're not cancerous. If they're non-cancerous, that means they're not going to break apart into little pieces, and those little pieces are not going to float to other parts of your body, which are going to cause cancer there as well. Those type of cancers are known as malignant tumors. Malignant tumors, when they reach blood vessels or lymph vessels, which are traveling through all the different parts of your body carrying fluids like blood or like lymph, if if just one of those cancer cells from a tumor gets into that bloodstream, it could be carried to any other part of the body. It's called as a metastasis. It will metastasize, meaning it's going to latch on to another part of the body, and it's going to grow a tumor there. That's why, if you think back to some people that you may know that have had cancers, it may start in one place, like it may start as testicular cancer or breast cancer or colon cancer, but then they say, oh, well, it spread to their heart or to their lungs or to their brain. So that's how cancer is spread. It's actually one teeny tiny little cell that breaks off from a malignant tumor and passes to other parts of the body. Okay. So uncontrolled cell growth. There's a really good video I really highly suggest, and if you're in my class, you're actually going to watch it, but if you're not in my class, I would highly suggest that you do watch it. Go search it on YouTube. It's called Dear 16-Year-Old Me. It's really powerful. It's about skin cancer and melanomas and how easy it is to check yourself. Me, as I'm very fair-skinned, I'm very pale, and I'm not saying that jokingly, I actually am, but so I'm more susceptible to skin cancer because I have less melanin protecting my nuclei of my skin cells, so I'm going to be more susceptible to skin cancer. So I need to check regularly for those little melanomas. But please, please, please watch the video, and please watch yourself, especially if you're thinking about tanning or spending lots of time in the sun. Make sure you're using sunblocks and maybe rethink tanning. Okay. But again, there's all kinds of really good videos out there about cancer and cancer cells. We're not going to take the time to do those right now. Okay. So, causes of cancer. This is obviously something that everybody wants to know. How do I get cancer? If I touch somebody with cancer, will I get it? Well, no, it's not passed on like that. Okay. Most of the time, the cancer is caused by a mutation in your DNA. It's just the luck of the draw, unfortunately. It could be caused by any of those things that we talked about when we talked about mutations, UV rays, chemicals, and radiation. If that happens, and especially if it happens to the gene that controls cyclin, and if it doesn't make cyclin or doesn't produce enough cyclin, then that cell is not going to stop reproducing. It's going to keep going through that cell division. So mutations in your DNA is the biggest one. Certain viruses can cause cancer. The human papilloma virus, HPV, causes cervical cancer. So please, please, please make sure if you haven't gotten a shot yet, you please go get that HPV shot to prevent cervical cancer, especially you females out there. Other chemicals, and then the other biggest one is genetics. Sometimes you are predetermined because of your history in your family. Sometimes you're predetermined to have certain types of cancer. So if you know of a history, especially of things like breast cancer or testicular cancer or colon cancer in your family, that you are getting yourself checked regularly, talking to your doctor, and staying up to date on the things that you should do to help prevent those things. Okay. So here's our list of important terms. I know it's a lot. I know we've covered a lot of things, but please take the time to make sure you know each one of these terms, be able to use it in a sentence, know the idea behind it, find ways to connect ideas, connect words, and if you can do that, you're going to be good to go on the test. Hopefully this was helpful to you. I kept it under 15 minutes. Whew. Have a great day. If you have questions, please don't hesitate to contact me at jkubuske@gocathedral.com. But please have a great day and stay safe out there.